Welcome to Biology at Priyojit, my dear viewers. Have you ever wondered how scientists produce human insulin using bacteria, or how DNA testing identifies criminals and solves paternity cases? All of this is possible because of one powerful technology, that is recombinant DNA technology. Today we will cover insertion of foreign DNA into vector, introduction into host, selection methods, genomic and cDNA libraries, PCR, DNA fingerprinting, DNA microarray, Sanger DNA sequencing method. So let's begin step by step. What is recombinant DNA technology or rDNA technology? Recombinant DNA technology is basically a method in which DNA from two different sources is joined together and introduced into a host organism to produce a desired product. Example, human insulin production in bacteria. So the tools required in rDNA technology are, think of it like a molecular surgery, a restriction endonuclease which acts as a molecular scissor, DNA ligase which acts as molecular glue, cloning vectors which are the DNA carriers, and competent host cell, usually bacteria like E. coli. So insertion of foreign DNA into vector. This process has three main steps. First one is the isolation of DNA. Second one is the restriction digestion with the help of the restriction endonuclease. Both donor and plasmid DNA are cut using the same restriction enzyme. And to isolate DNA, we isolate the DNA from donor organism, and we also need vector DNA, usually plasmid. Most common plasmid comes from E. coli. And the third step is ligation, where DNA ligase enzyme joins the foreign DNA with the plasmid. Here enzyme used is T4 DNA ligase, and now we get the recombinant DNA or rDNA. So introduction of the vector recombinant DNA into the host cell. This process is called transformation. A host organism can be used as E. coli bacteria. So calcium chloride treatment increases the permeability of the bacterial cell, plus heat shock also increases the permeability of cell membrane. Electroporation and microinjection is performed in case of eukaryotes. Electroporation is done in prokaryotes. So now bacteria contain recombinant plasmid. So treat foreign DNA and plasmid with restriction enzyme and DNA ligase. Then introduce the recombinant plasmid into cultured plant cells. Then regenerate new plant from the cultured cells. Selection of recombinant bacteria after transformation. Not all bacteria contain recombinant DNA, so we need selection method. First one is the antibiotic selection. Plasmids contain antibiotic resistance genes. Example, AMP-R, that is ampicillin resistance, or TET-R, that is tetracycline resistance. So only bacteria containing plasmid will survive on antibiotic medium. So limitation, it cannot differentiate recombinant from non-recombinant plasmid. So this is the image showing the antibiotic selection procedure. Now the second procedure is the blue-white colony selection, which is based on LAGZ gene and beta-galactosidase enzyme. Here the substrate is XGAL is used. Mechanism: if LAGZ is intact, blue colonies are formed. If foreign DNA is inserted into the LAGZ, then LAGZ will be disrupted and then white colonies will be formed. So white colonies indicate the recombinant bacteria and blue colonies is the non-recombinant. So we must select the white colonies. Genomic library, a collection of cloned DNA fragments representing the entire genome of an organism. So steps are: isolate total DNA, partial restriction digestion, insert into vectors, transform host, and each clone carries different DNA fragment. This is the construction of genomic DNA library. Here double-stranded DNA is cleaved with restriction nucleates, then genomic DNA fragments are produced, then they are inserted into fragments into plasmid, then recombinant plasmids are produced, then it should be introduced into plasmids into bacteria, then genomic DNA library are formed. cDNA library, this is the collection of DNA fragments made from mRNA. Here the isolation of the mRNA, then use reverse transcriptase to make cDNA, then make double-stranded DNA and then insert into vector and transform the host cell. Here is the RNA extraction. and then RNA extraction is performed from the human cell. Here is the RNA, and then cDNA synthesis, amplification, and library preparation, that is cDNA library. So what is the difference between genomic library and cDNA library? Genomic library contains introns, but cDNA library has no introns. Genomic library has regulatory regions, but cDNA has no regulatory regions. Genomic library represents the whole genome, but cDNA represents only expressed genes. Screening of libraries. After library construction, we must find the desired gene. First of all, the technique is colony hybridization, which is used for plasmid libraries. The steps are transfer colonies to membrane, the lyse cells, and add labeled probe, then hybridization, then which is followed by autoradiography, and then positive colony shows signal. Plaque hybridization, this is the image showing the colony hybridization procedure, and plaque hybridization here is the used for fudge libraries. Example vector is bacteriophage lambda. Similar procedure, but screening fudge plaques instead of bacterial colonies. Now we should go for the polymerase chain reaction, which was developed by Kary-Mullis. It's an in vitro amplification of specific DNA sequence. For this processes, we require template DNA, primer, the DNTPs, Taq polymerase, magnesium 2+ ions, and thermal cycler. So Taq polymerase comes from a bacteria, Thermus aquaticus, the T from Thermus and AQ for aquaticus, that is Taq polymerase, which is a heat-loving bacteria. So steps of PCR include the step 1 is the denaturation press, occurs at 94 to 95 degrees Celsius. Step 2 is the annealing process, which occurs in between 50 to 65 degrees Celsius. And step 3 is the extension process, which occurs in 72 degrees Celsius, 72 degree. So each cycle doubles DNA. After n cycles, 2 to the power n copies of DNA will be produced. So this is PCR or polymerase chain reaction. DNA fingerprinting was developed by Alex Jeffries based on VNTR, variable number tandem repeats, and the steps are here: the isolation or extraction of DNA, then restriction digestion, then gel electrophoresis, then southern blotting, and then hybridization. So it is applied for forensic science, paternity testing, criminal identification, and population genetics. DNA microarray. The process is used to study thousands of genes at once. Principles include hybridization between fluorescent cDNA and DNA probes on chip. The steps include extract mRNA, then make fluorescent cDNA, laser scanning, and then data analysis. So it is applied for cancer diagnosis, gene expression analysis, drug response study, etc. DNA sequencing or by means of Sanger method, developed by Frederick Sanger, also called as chain termination method. Its principle uses normal nucleotide or DNTPs, chain terminating nucleotides or DDNTPs, and DDNTP lacks 3' OH group, so DNA elongation steps. And the steps include template DNA, primer, DNA polymerase, 4 reactions with DDATP, DDTTP, DDGTP, and DDCTP, then gel electrophoresis, then read sequence. Here the modern method uses fluorescent automated sequencing. So in conclusion, restriction enzymes and DNA modifying enzymes are the core tools of genetic engineering. Restriction enzymes precisely cut DNA at specific sequences, while modifying enzymes like ligase, polymerase, and nuclease join, synthesize, and refine DNA molecules. Together they form the foundation of recombinant DNA technology, enabling gene cloning, sequencing, and modern biotechnological advancement. So keep exploring the powerful tools of molecular biology. Thank you.